In this video, we are looking at microscopes as tools for looking at cells. Most things at the cellular level are way too small to see without a microscope. So the human eye can see down to close to almost 100 micrometers, but not quite. So range of a human eye, something thinking about frog eggs, you may have seen those in a pond, get a little lower than that, but you're kind of limited in what you can actually see. Most plant and animal cells are definitely outside of that range, as are most bacteria. And you notice that there are different microscopes that can detect some of those things and some that can go down even smaller. So we are going to be looking at light microscopes and electron microscopes. Light microscopes are used to view an entire cell, and one of the benefits of them is that you do not have to kill the organisms. You can actually view living organisms. Electron microscopes require that the sample, the organism, is killed and affixed to a surface in order to be investigated. And they do, though, the benefit of an electron microscope is that they can view cell parts, so they can get down to some really tiny details and some things that are really small. So, for example, atoms are your large atoms, your high weight atoms are in the zone of about 1 angstrom in terms of their size. And that is extremely tiny. So, a transmission electron microscope could be used to view something like that. Well, something like looking at proteins or viruses, a scanning electron microscope might be able to provide some good insight into that. So let's look at transmission electron microscopes. They are used for seeing internal cell structures. So again, the sample is actually affixed. Um, Electrons are transmitted through the cells, and the rate of transmission is actually what the microscope interprets to then produce an image. So it's not actually seeing the image, it's reading what the electrons are doing in order to create an image. Um, the images are black and white, but a lot of times they'll appear in color because they are later colored using some different techniques with programming that recognizes certain frequencies of those electrons and colors them certain colors. Just allows you to differentiate cell structures or tiny structures a little more clearly. Um, Compared to a light microscope, a transmission electron microscope allows us to view very tiny things, so the very smallest things looking down into the realm of atoms and molecules and proteins and viruses. Both of our types of electron microscopes can do that. Again, the transmission electron microscope actually sees through cells, and we're going to see that's different than our next microscope. Here's an example of what a transmission electron microscope image could be produced. This is the coronavirus, and so viruses are very tiny. You can see some of the external structures on the outer surface of that virus. You can see it has some internal structures as well. Again, this image was actually scanned black and white, but computer programs are able to recognize differences in those frequencies, the transmission frequencies, and color them differently. Another type of electron microscope is a scanning electron microscope. It's different from the transmission electron microscope in that the transmission, remember, actually goes through cells. It looks through cells. A scanning electron microscope, as scanning suggests, it actually just reveals surface details. So it's bouncing electrons off. As the electron bounces back, it gives a certain sort of wavelength, and that is interpreted by the scanning electron microscope. Again, these images are black and white, but they can be colorized, which makes them even a little more interesting to look at. Again, they have the similar range to it is the same range as the transmission electron microscope. It's just used differently. Again, disadvantage of scanning electron microscopes, extremely expensive, difficult to operate, and you must kill the organism in order to view it. These are pollen grains of various different types of plants as seen under a scanning electron microscope. So they do reveal some really interesting images. And I have to say myself, this is one of my favorite microscopes, the scanning electron microscope, because it really kind of reveals the true extent and fascinating nature of the world around us. Finally, we have light microscopes. We have two types of light microscopes. We have a compound light microscope and a confocal light microscope. They do not have the magnifying power, as you can see by our scale here. They see down not quite to the level of viruses, but they are useful for looking at bacteria, archaeans, and most plant and animal cells, and even some larger things. So you might even use a scanning light microscope to be able to just let's say you're looking at, you want to look at a cricket up close. You might just want to look at the surface features of the cricket and see them slightly magnified. So you could use a certain type of light microscope for that. And I've looked at a lot of insects under these various different microscopes. And I have to say, it's very fascinating. So they work by transmitting light through cells. So again, you can use them for actually looking at living cells. And if you do an onion root tip mount, we may do that lab at some point in time, you can actually see cell division actively happening during the viewing process because those cells do not have to be killed for that to occur. The top left image is a compound light microscope, so that might be something that you see. So notice that it doesn't, it is literally kind of what you would see if you were looking at this with the naked eye. It's just greatly enlarged. And you can see that the light is passing through the organism. So the different amounts of light that pass through make something look lighter or darker. So these are tiny little single-celled organisms, and you can see their internal compartments and things like that. I believe those are paramecia, by the way. The bottom right is with a confocal light microscope. It actually passes a laser through the cell, and the laser brings back the image by the amount of light that is transmitted through the substance. Often these are used with fluorescence, and that is what you're seeing here when you're looking at the foregut and midgut junction of an insect.